Well, thanks everyone for joining me today. My name is Nate Russart. I'm a principal scientist here at Aldevron. I'm here to talk to you today about our alchemy cell-free DNA technology and how you can use this technology in your mRNA manufacturing solutions. First, a little bit about Aldevron. So, we were founded in 1998 on campus at North Dakota State University here in Fargo, North Dakota. And since then, we've been partnering with our clients to advance their therapeutic applications, advance their capabilities, their capacities, and so on and so forth in order to get their programs to the clinic and to the commercial stage. All right, so we are primarily a custom manufacturing. So we operate across three major business units. Our plasmid DNA, this is where we got our start and did the bulk of our business still, where we produce traditional plasmids, both supercoiled and linear forms. We have next evolution constructs, including our nanoplasmid vectors. What I'm going to talk to you about today is our synthetic linear DNA technology, and then our proprietary upstream and downstream processes for E. coli-based manufacturing. On the RNA side, we produce that linear template. All of our IVT and capping enzymes we do produce as well. We're able to fulfill drug substance and drug product manufacturers for you with your constructs. On the protein side, we provide a number of custom protein manufacturing solutions for you, as well as some off-the-shelf CRISPR nucleases and other enzymes. All of these are available across research-grade GMP and GMP source manufacturing. Along with our custom manufacturing, we do offer a number of catalog solutions. In many cases, these are available in stock and ready to ship at a moment's notice. So once again, on our CRISPR nucleases, we both have wild type and some higher fidelity versions. Our IVT enzymes and capping enzymes that we use in-house and we offer to our clients as well. We have a number of viral vector plasmids that support your AAV or lentiviral programs, such as helper or RepCap plasmids. We have a number of plasmid backbones for either your DNA needs or your RNA manufacturing needs. And then we have a number of DNA and RNA reporter sequences as well. A number of these do have a drug master file on record to streamline your FDA filing. All right, so if we take an overview, we have our custom manufacturing, our off-the-shelf products, and then our workflows where we can really go sequence to vial. We're really there to work with you and offer you solutions for your entire drug program. As I mentioned, we are located in two areas. Our headquarters is in Fargo, North Dakota. This houses our plasmid DNA manufacturing, our RNA manufacturing, and all of our GMP facilities. Our Madison site is ISO certified, and this is primarily does all of our protein manufacturing, protein R&D, things like that. On the top left, we'll see this is an overview of our breakthrough campus in Fargo. It's a little hard to see from the picture, but there are basically two large buildings that are connected here. Down the center of those buildings is all of our GMP manufacturing suites, surrounded by support areas, support labs, and office areas. All right, so along with our DNA, mRNA, and protein businesses, we have a number of support services, including assay development, process design and development, technology transfer, stability testing, regulatory support. Also, we are part of the Danaher Life Sciences family of brands, and we see a number of familiar names down here on the right. So we're able to partner with these companies to really offer the full suite of support that we can to meet all of our clients' needs. And so that leads us into what we're here to talk about today, specifically our alchemy cell-free DNA technology. And so this is an offering that we launched in January of 2025, and at the research grade scale with plans to launch at the GMP scale later this year. Okay, so when we think about cell-free DNA technologies, the biggest advantage of this is the reduction in timelines. So if we think of a traditional plasmid-based workflow, we have starting material come in, we have to transform that into cells, streak plates, pick colonies, screen starters, grow that up, purify, then go into a linearization reaction, purify, and a QC. All right. For a cell-free workflow, we can take the exact same starting material that you would provide for a plasmid workflow, put that right into our amplification reaction, and then linearize one purification step and return it to the customer. So, the processing side is much faster. Once again, this is on the research grade side. Once we get into the GMP side, there's some advantages around removal of cell banking steps as well that will add to the time savings there. So we developed our Alchemy cell-free DNA technology for the production of mRNA. So this wasn't a material that we produced as a DNA standalone product and decided we could use it for mRNA as well. So as I said, we can take your starting material, we put it right into a rolling circle amplification reaction. So this is an isothermic reaction, which means it does scale really well, and we can go to large scale with this. Following that is linearized with a restriction enzyme, very similar to you would with a plasmid DNA construct, and we can in fact use the same linearization enzyme. We would purify, and then it can feed right into an IVT reaction, the same as a linearized plasmid DNA would. We do use the exact same plasmid that you have been using, so we don't involve any retrofitting of that construct into a proprietary backbone. So there's no cloning steps involved. I will say we do start from a plasmid, so we don't start from fully synthetic material, and that's just because there are some issues around the fidelity of fully synthetic material that we want to stay away from. So what our feature looks like when it's all said and done is it's a linear double-stranded piece of DNA. There's no closed-ended steps. And in fact, we never have any closed-ended steps in our process. So there's no need to decap these ends or have an assay to show that there's a decapping has been complete, and no risks to double-stranded production with that end is still in place. And then once again, the same exact sequence that you would have from a linearized plasmid. So we do have that backbone in place where that is a little bit different than some of the other solutions that are on the market. But this allows you to very easily swap out your current plasmid DNA workflow, the cell-free construct instead of the plasmid. Also, if you have assays that target areas of that, you won't have to change anything there. That sequence is still there and able to be targeted. So once again, you're able to go directly from a plasmid DNA workflow into the cell-free DNA workflow. That's how we've designed this and came up with this product. And so, as we were developing this, we worked with a number of our collaborators, and we offered them both our cell-free DNA and a linearized plasmid DNA that is produced through our research-grade manufacturing operations here. Gave them both constructs, manufactured different ways, the same exact sequence, and asked for them to evaluate them in their hands and provide us feedback on how it worked. So, and we offered no advice on what to do in the case of cell-free. They use it directly into their current workflows. So here's some data we have from a collaborator. So they had two constructs. Our graphs here, the orange bars represent the linear plasmid DNA template, and then also the mRNA that was produced from the pDNA template. The yellow bars represent the linear cell-free DNA, and then the mRNA that was produced from the cell-free DNA. All right, so where they measured the integrity or the amount of full-length product by capillary gel electrophoresis. And so we can see across both constructs that there's really no difference in the integrity of the template starting material. And then when we look at the mRNA that was produced from both of these, we can see that in this case, the mRNA produced from cell-free DNA, the integrity was a little bit higher. I wouldn't expect this to always be the case. What we do typically see is that it's very similar. In this case, with this client, their IVT workflow, it may have worked a little bit better. Still looking into that, but I would expect you to see very similar levels of integrity from mRNA produced from both plasmid DNA or cell-free DNA template. Okay, so once again, this fits very easily into your current IVT process that you already have. So we had another collaborator that was interested if the cell-free template would be removed from the IVT reaction through their DNase treatment, as well as plasmid DNA template. So once again, this was their IVT process that they had developed and optimized for plasmid DNA. The DNase treatment was developed and optimized for plasmid DNA. They did nothing different except for swapping out the template from the plasmid DNA to our Alchemy cell-free DNA. And so what we can see is that the levels of residual template after treatment were, in this case, our cell-free template than they were on the cell-based or the plasmid DNA template. Once again, I wouldn't expect this to always be the case. It happened to be so here. The other thing I want to point out here again is that they did not have to develop a new qPCR assay to measure the residual levels of plasmid DNA. Their assay targeted, part of that plasmid backbone that they've already been using, and so that backbone was still in place, and so they could use the exact same sets of primers and qPCR conditions to measure the residual template. Okay, last but not least, so in some ways, looking at expression is kind of the table stakes. Like, it has to perform as well as plasmid DNA. If it wouldn't, then it wouldn't be an acceptable product, right? But at the end of the day, this is the most important thing, right? So here we have a client that was producing self-amplifying RNA. So this does work just as well for long constructs as well as shorter constructs. In fact, I would argue that it's probably going to be, as the construct gets longer, it's going to be more difficult to produce the mRNA than it is the template for it. But they were producing a self-amplifying RNA for a vaccine program. And so the expression of an antigen in HEK cells by flow cytometry. And they saw very similar levels of expression from mRNA produced from plasmid DNA template as well as cell-free DNA template. And so, so far I've covered all the very positive things about cell-free DNA and what it can do, but this is a newer technology, and there is still some space for it to improve. And so there are some potential drawbacks to it, and so I want to highlight those as well, so that we can make sure that we're offering a product that works for your program. And if it is not acceptable for you, then we also have the pDNA that we can offer as well. And so you'll hear fidelity come up a lot when we talk about cell-free DNA. So we do use a Phi-29 polymerase. It's a very high fidelity polymerase. However, it doesn't have the same mechanisms that E. coli cells have in order to error correct and really have the high fidelity. Those plasmids in E. coli are kind of the gold standard right now. And so anytime a new product comes out, usually we want to improve on everything. And so in this case, we know the fidelity is not going to be as high. What does that mean? We can talk a little bit about that, but just to set the stage a little bit and make sure we're on the right track. When we talk about fidelity, we talk about the rate of incorrect bases being included into individual DNA strands. Okay, if we do Sanger sequencing or build a consensus sequence, we're going to get 100% match to the reference sequence. All right, we're not talking about these SNPs or variants that are going to be present in everyone. It's going to be these individual nucleotides that are going to be changed. So in the example here on the right, we have 8 DNA strands. If we did the consensus sequencing for this, it would be 100% match, but we do see in that fifth sequence from the top, there is that one mutation there from an A to the C. So what is the rate of that? That's what we want to look into. And if you look into the 529 polymerase, you'll see a published error rate of 1 times 10 to the negative 4 to 10 to the negative 6. That's quite a big range. But that's the polymerase itself, not the entire reaction that we're working with. So it's very difficult to measure these low error rates. And so I'm going to go into a couple of methods that we've used to look into that here. So first, we did some NGS sequencing, some Illumina sequencing. We think about things like Illumina and PacBio, HiFi. We think of very high accuracy. Even though they're very high accuracy, when we're looking at very low error rates, they're not accurate enough. And so there needs to be some specialized methods involved and to really tease out the errors that are introduced through the sequencing process or through the library preparation process. And so one method to do that is using these unique molecular identifiers. And so we did this. And how these work is we tag individual strands of the DNA with these random barcodes, usually like 8 mers. And then after we do the library prep and amplify those, we're able to take all of the reads that come from those individual strands by the barcode and see if they are in agreement with each other or not. And so in the example on the right here, if we look at the top set in the blue box, all of these reads can be traced back to the original molecule by that blue barcode. All right. And then we see these green circles represent, a change from the reference. Now we see that change in all 3 of those reads. That means it was probably there in the original DNA strand. And so that's a true error. Now, if we go down, and let's say to this green box, we see, once again, 3 green circles that are all in the same place. That means that mutation was in the original DNA strand. But we have that red one, which is only present in 1 out of the 3 reads. Now, that means that was probably a sequencing-induced error and not something that was present in the original strand. And so using this method, we're really able to tease out a lot of the noise, background noise, or the errors that are introduced through sequencing to really try to hone in and identify what that true error rate is. Some of the drawbacks on this is that it requires a lot more coverage of the sequencing. And also, there's still some error that's present. And so it's good to compare it to, what the standard is now, which would be a plasmid. And so we did partner with some of our colleagues over at IDT, one of our sister companies through Danaher, to look at the error rate of our alchemy cell-free DNA using this UMI process. So one thing I will point out is that we have a G to the A mutation and C to the T mutation, which are very similar things, just opposite strands. And this is a common mutation due to the nature of those nucleotides and how they're built. All right, so we weren't, really happy with the NGS workflow that we used and the results we got because there was still a high error rate there. So we switched to a little more of an old school method, but this method shows a phenotypic change, and it's really hard to argue when you see the bacterial colonies functioning differently. You can get sequencing data back, and sometimes it's hard to believe these numbers and things that you get in these sequences, but it's really hard to argue when you see a difference on a plate. And so this is just an example here, but a bit of a refresher. We use this LAC-Z blue-white screening method. So this is used, very, very frequently for cloning. And so the idea is that there's this LAC-Z gene in the genome, and it's incomplete. And in the plasmid, we have this LAC-Z alpha fragment. And then if that fragment is made completely, it will allow the beta galactosidase to be active, turning X-gal blue. And so if that laxi alpha is intact, the colony will be blue. If it's not intact, it'll be white. So when you're cloning, you're trying to clone inside that laxi alpha fragment, and you're hoping to get a white colony. That means that whatever you cloned in there is right. As we look into this for fidelity, we want a blue colony to be present. That means that that laxi alpha was copied correctly and it has the correct sequence there and is able to make that functional protein. What's really nice also about this laxi alpha fragment is it's very well characterized. And so any mutation, any possible mutation that can happen there, it's known whether it is a loss of function mutation or not, or just a silent mutation. Okay, so we use this method. Once again, we did compare to plasmid DNA, but we amplified some DNA, and then we had to recircularize it in order to put it back into the cells. And we plated, almost 12,000 colonies of both our plasmid DNA and our cell-free DNA. We do see, quite a difference in the number of mutant colonies. So these colonies that turn out white or even a light bluish color sometimes. So with plasmid DNA, we had 2. With the cell-free DNA, we had 52. So about an order of magnitude difference. Now, because we know very well with that, the characterization of that laxi alpha fragment and which mutations are detectable or not, there's a formula where you can plug these numbers into, and it'll give you an error rate. So it's not looking at the full length of the sequence, just, again, those detectable mutations. And so from plasmid DNA, we get an error rate in the order of 7 times 10 to the negative 7. And from our cell-free, we're at 2 times 10 to the negative 5. So if we subtract out the plasmid DNA as the standard, the error rate from cell-free DNA is about 2 times 10 to the negative 5 higher than plasmid DNA. We were also able to look at these mutant colonies, as well as some of the colonies that weren't mutated and still had their functional proteins, and sequence those just to ensure that our results were in fact real, right? So that we were seeing actual mutations in these white colonies, and in the blue ones we weren't. And so we did find mutations in all of the white or light blue colonies. In the blue colonies that we sequenced, we did find one mutation, but it was one that was a silent mutation, and so it made sense that we didn't see a difference there. And so I think our sequencing results of these mutant colonies, agreed with the results that we got here, and I like these numbers a little bit better. We'll see even the plasmid DNA, the error rate is not as high as what you might see in the literature for plasmid DNA. But oftentimes that error rate is per round of replication, not in your final product. And so as we know, during a fermentation, plasmid is replicated many, many, many times. And so an error introduced early in that might show up a lot more later. We're looking at just the total error rate of the final product, not per round of replication. Okay, the other thing I wanted to touch on when it comes to our cell-free DNA and generally from cell-free DNA, when we talk to clients and customers who have been evaluating different materials, is that you tend to see higher levels of double-stranded RNA from cell-free templates that are out there. And so here we can see, the first 2 bars represent plasmid DNA that was, mRNA that was produced from plasmid template using a wild type T7 polymerase. And the next bar is our cell-free template, also with the wild type T7. So, a little over 2 times higher amount of double-stranded RNA with this construct. What we tend to see is somewhere in the range of 2 to 4 times higher amounts of double-stranded RNA that's produced. As you all know, double-stranded RNA, it's a difficult thing to quantify depending on your standards and your methods that are being used. So we try to just reference the plasmid as the control here. But, These next two bars represent we have a mutant T7 polymerase that's designed for lower double-stranded RNA production, which we call high-cap polymerase that we've licensed with Codexis. And we see it is really effective at dropping the amount of double-stranded RNA that's produced, both from plasmid and from our cell-free. And so, in this case, our cell-free DNA template partnered with the high-cap polymerase has lower double-stranded than a plasmid DNA with wild type. And so, this is kind of up to you to understand and decide what the levels of double-stranded RNA that your program requires is, and how to mitigate those. We recommend partnering it with the high cap. There's also downstream purification approaches and things like that can be used to decrease those amounts if it doesn't fit your needs right now. And as always, you can also go back to our plasmid DNA as well if this doesn't fit your needs around double-stranded RNA or around the fidelity. And so some of the outstanding questions, if you're thinking about converting to cell-free DNA from pDNA, what level of fidelity is acceptable? This might be dependent on the modality, whether you're using a vaccine or some kind of gene editing approach. What is the safety risk? What is the regulatory risk? There's not a lot of guidance on that right now. And then once again, what levels of double-stranded RNA are acceptable? It could be dependent on modality again, whether it's a vaccine or a therapeutic, depending on what your dose size is. How are we minimizing double-stranded RNA through other purification methods or IVT conditions? And also, how are we measuring it? And so in summary, I just want to say again, we've launched our Alchemy cell-free DNA technology this year at the research grade quality level. Very excited about it. You can send in your starting material just as you would with plasmid. We can amplify it, linearize it, same enzyme. Feeds right into an IVT process. Looking to launch GMP later this year. And we have the capabilities to go really sequence to vial here partnered with our LNP encapsulation, our fill finish, and our drug product pipeline. And so I appreciate you taking the time to listen to this with me today. If you have any other questions or you want to learn more, feel free to reach out to me. My email address is listed here. And thank you once again for taking the time.